Hello students, myself Ajay Kumar Shivastav. I am senior faculty in Botany in Sri Chaitanya Gosala campus. In the previous lecture, we studied about the structure of nucleotides and we said that nucleotides polymerize to form the nucleic acids. The nucleic acids are of two types, DNA and RNA. DNA was accepted as the putative genetic material. Putative means universally accepted. So, it was universally accepted as the genetic material. So, now let us see detailed structure about the DNA and how it was discovered. Okay. So, DNA or deoxyribonucleic acid, this was discovered by Friedrich Mischer from the pus cell nucleus, and since it was found in the nucleus, he called it nuclein. But later on, seeing its acidic nature, it was named as deoxyribonucleic acid. Okay, fine. Next, Wilkins and Franklin, they performed X-ray crystallographic experiments and they proved that DNA is a helically coiled structure. Fine. Next, Watson and Crick in 1953, with the help of the work of Wilkins and Franklin, they gave the detailed structure of the DNA and they said that DNA consists of two polynucleotide chains that are bound to each other, that are linked to each other, and it is helically coiled in right-handed direction. This is called as right-handed model of DNA or B form of DNA. Okay. So, Watson and Crick and along with Wilkins, they were awarded Nobel Prize in 1962 for the discovery of the structure of DNA. It was thought to be one of the major discoveries of this century. Okay. Watson and Crick also got help from the work of Erwin Chargaff. There was a scientist called Erwin Chargaff. He studied double-stranded DNA and he gave two general principles. He said for a double-stranded DNA, the amount of purines is always equal to the amount of pyrimidines. It means adenine plus guanine is equal to thymine plus cytosine, or adenine plus guanine upon thymine plus cytosine will always be 1. This was the first rule. Second rule was that it is not necessary that in a DNA number of AT pairs and number of GC pairs are equal. So, A plus T upon G plus C, this varies from species to species, but for a particular species it is constant. Okay. So, A plus T upon G plus C is constant for a species, but it varies from species to species. Generally, this ratio is less than 1 in prokaryotes. As you can see in the bacterium Escherichia coli, it is 0.92, and in eukaryotes it is usually more than 1. For example, if you see in human beings, it is 1.52. Now, we are going to see the structure of DNA, and that is bDNA. Actually students, DNA exists in many forms, okay, but the most common and the most stable form is the B form of DNA. Other forms are aDNA, cDNA, ddNA, zDNA. These all other are called as topoisomeric forms of DNA, and these are temporary forms. And Watson and Crick gave the structure of this bDNA. So, let us see what are the salient features. First, they said that the DNA consists of two polynucleotide chains that are held together and they are helically coiled. So, consist of two helically coiled polynucleotide strands. Second thing, they said that this coiling is in the right-handed direction, that is why it is rhDNA, right-handedly coiled DNA. Okay. Next, one complete turn, or we call it as pitch, that is 3.4 nanometer or 34 angstrom, and in one complete turn there are 10 base pairs. Okay. 10 base pairs in one complete turn. It means the distance between the adjacent base pairs is 0.3 nm or 3.4 angstrom. the diameter of DNA double helix is 2 nm or 20 angstrom. Next point is that the two strands are complementary to each other. This means that if we know the nucleotide sequence of one strand, we can predict the nucleotide sequence of the other strand, and this feature is very important for replication of DNA. It is due to this feature that when a DNA molecule makes its copies, both the daughter DNA are exactly similar to the parental DNA molecule. Okay. Next feature is the polynucleotide strands have backbone of sugar and phosphate, sugar, phosphate, they alternate each other, okay. And the nitrogenous bases are at right angle, they are projected inwards at right angle to the backbone, okay. Next is the two strands are held together by hydrogen bond, okay. So, the nitrogenous bases of the two strands, they are joined by hydrogen bonds. So, the two strands are held together by hydrogen bond. Now, adenine always pairs with thymine or vice versa, thymine with adenine with two H bonds, okay, with always you can see two H bonds, and guanine always pairs with cytosine or vice versa, that is cytosine to guanine with 3 H bonds. Now, this ensures that a base pair is always between a purine and a pyrimidine. Okay. So, remember purines have 2 rings and pyrimidine has 1 ring. It means a base pair will always have constant 3 rings, either purine is this side or pyrimidine is this side. Okay. This ensures that the distance between the two strands is uniform. See, okay. So, always the distance between the two strands is uniform, it is due to this, okay. Next is the plane of base pairs stack, because it is not straight, you see it is helically coiled. So, when it is helically coiled, what happens? One base pair is stacked over the other, okay. This provides stability, and another stability is provided by this hydrogen bonding. So, this hydrogen bonding and stacking of the base pairs one over the other, this provides stability to the structure of the DNA. Now, I will show you the diagram where we can see all these features in the diagram. Now, let us see the diagram to elucidate those points. Okay. As we said that it consists of 2 polynucleotide chains that are helically coiled, that we can see here. Okay. We said that there are base pairs in this, and the length of one complete turn or one pitch is 3.4 angstrom. The distance between two adjacent base pair, 3.4 nanometer. Distance between two adjacent base pair is 0.34 nanometer, and the diameter is 2 nanometer. Okay. This we can see here. Other features we can see here. So, I have just tried to show other features. You see, we said that the two strands are complementary, means that if we know the nucleotide sequence of this strand, we can tell. Why? Because adenine will always pair with thymine and guanine will always pair with cytosine. So, if we know the nucleotide sequence of the one strand, we can predict the nucleotide sequence of the other strand. This is the complementary nature. Next point, we said that in both the polynucleotide chains, there is backbone of sugar and phosphates. You can see the backbone of sugar, phosphate, sugar, phosphate, sugar, phosphate. Okay. And the nitrogenous bases are projected inwards. This point we can see here. Okay. Next, we said that the two strands are held together to each other with the help of hydrogen bonds. So, these purple color, these are showing the hydrogen bonds. You see, between adenine and thymine there are 2 hydrogen bonds, and between guanine and cytosine there are 3 hydrogen bonds. It means always a purine is pairing with a pyrimidine or vice versa. As a result, the distance between the two strands is uniform throughout its length. Okay. This point is also clear. Next, we said that the two strands are anti-parallel, means equidistant but opposite polarity. Then we can also see that in this strand at this end there is 3' end and here there is 5' end. But in the complementary strand, here there is 3' end and here there is 5' end. So, all the features that we have discussed, we can see in these two diagrams of DNA. Now, very soon after the discovery of the structure of DNA, Francis Crick, he proposed a scheme to show the flow of genetic information, and this scheme is called as central dogma of molecular biology. He said that DNA, which is the genetic DNA, it can replicate to make its own copies. Okay, DNA can replicate to make its own copies, and this DNA by the process of transcription can form a type of RNA called mRNA containing the message of DNA, hence called as messenger RNA. Now, what is this message? This message is that which type of protein will be synthesized, and then by the translation of this coded message present in mRNA, a specific type of protein is synthesized. So, this flow of genetic information is unidirectional from DNA to mRNA to protein, okay? And in most cases it is correct. But later on certain viruses were found where a reverse flow of genetic information was seen, that they can synthesize DNA from RNA. Okay. The enzyme which was involved in this was called as reverse transcriptase. The phenomena was called as reverse transcription, and after the name of the discoverer, it was also called as Steminism, because Stemin and Baltimore discovered this phenomena, and the resultant DNA that is formed by the reverse transcription, it was called as cDNA, means complementary DNA. So, this is an exceptional case where a reverse flow of information takes place in certain viruses, that is from RNA to DNA, and these viruses are called as retroviruses, and a very good example is HIV. Now, you see, we said that DNA is a large biomolecule. In both prokaryotes as well as eukaryotes, the length of the DNA that is present in a cell, it is much more longer than the diameter of the cell or the nucleus itself. The question arises, then how such a long DNA gets accommodated in a small cell or a small nucleus? Children, this is possible due to a phenomena called packaging. But first of all, let us see, can you imagine what would be the length of the total DNA which is present in one cell of us? Okay, in one cell of us, what could be a cell that we cannot even see by naked eyes, the DNA present, what is the length of that DNA? Let us see. So, in a typical mammalian cell, when we say we are talking about our own cell, okay, the total number of base pairs present is 6.6 into 10 to the power 9, okay, because one set of chromosome has 3.3 into 10 to the power 9 and a diploid cell has two sets of chromosome, okay. And you know that the distance between two consecutive base pairs is 0.3 nm, thus we have already calculated, or you can say 0.34 into 10 to the power minus 9. It means the total length will be this multiplied by this, and this comes to around 2.2 meter. It means in one of our cells in the nucleus, the DNA length is total 2.2 meter, while the diameter of that nucleus is just 10 to the power minus 6 meter, or you can say 1 micron. So, in a nucleus of 1 micron diameter, 2.2 meter long DNA is accommodated. This is possible by packaging, and it is not that only in eukaryotic cell. Let us see prokaryotic cell also. You know, a bacterial cell is very small, very small, but in a bacterial cell, the total length of DNA is 1.36 millimeter, which is much larger than the size of the bacterial cell itself. Now, let us see how many base pairs can be present in this 1.36 millimeter long DNA. So, 1.36 millimeter, if we convert into meter, it will come 1.36 into 10 to the power minus 3, okay, into 10 to the power minus 3, okay. And the number of base pairs we can calculate by dividing it with the distance between two adjacent base pairs. So, we are dividing 1.36 into 10 to the power minus 3 by the distance between two adjacent base pairs. This we know 0.34 into 10 to the power minus 9, and this comes 4 into 10 to the power 6 base pairs. But here also children, although bacteria do not have a true nucleus, but it does not mean that this DNA is scattered throughout the bacterial cell. This DNA is confined to a specific region called as nucleoid, and it is packaged with the help of certain basic proteins that form loop-like structures, and these proteins, they have positive charge while DNA has negative charge. As a result, it is held with the DNA. Now, let us see the DNA packaging in eukaryotes. So, it forms repeating units, and this unit is called as nucleosome. Okay. A typical nucleosome consists of two parts, a histone core which is an octamer, and around it the DNA is wrapped. So, you see there are two parts, histone core or histone octamer, and second part is DNA. Now, histone are proteins that are rich in basic amino acids like lysine and arginine. So, they have basic nature, basic proteins, and these amino acids have side chains which are positively charged. Okay. And this histone core, why I am saying octamer, because this core consists of two molecules each of 4 types of histone, H2A, you see H2A, H2B, H3 and H4, 2 molecules each, octamer. There is one 5th type also, but that is present outside, not in the core. Okay. So, this is the histone. Now, around this histone DNA is wrapped, and how it is so tightly held? Because we said that these amino acids, they have positive charge, and DNA, due to its phosphate group, it has negative charge. So, due to this opposite charge, the DNA is very strongly held, wrapped around the histone core to form the nucleosome structure called nucleosome. And as we can see, a nucleosome contains 200 base pairs long DNA wrapped around the histone core, and outside there is another histone H1 which is present outside them. So, you can see this is the histone core which is the octamer, and around this histone core you see this orange color, this is the DNA which is wrapped around it, and outside, to make this DNA tightly held, this is H1 histone. This is a single nucleosome. Now, repeating units of nucleosomes are present which forms a structure called as chromatin, and when seen under the electron microscope, this appears as a beads on string structure that we will discuss now. Okay. Now, let us see what is chromatin. We said the unit of packaging is nucleosome, and a string of nucleosomes is formed which is called as chromatin, and when we will see under the electron microscope, this string of nucleosomes or this appears as beads on string structure. It appears as beads on string structure. Like there is a string at which at regular intervals beads are present. So, this array of nucleosomes that forms a chromatin appears as beads on string structure. Now, this chromatin further undergoes condensation. Okay, it further undergoes condensation, and maximum condensation you can see is seen at the metaphase stage of cell division that you have studied in cell cycle and cell division. So, maximum condensation of chromatin, it results in structure which we commonly called as chromosomes. It means chromosomes are formed by the condensation of chromatin, and chromatin is made up of repeating units of nucleosomes. Fine. Now, as I said, this chromatin further undergoes condensation and coiling to form chromosomes. So, for this higher level of coiling, along with histone proteins, certain non-histone proteins are also required. These are a group of proteins collectively called as NHC proteins, non-histone chromosomal proteins, like these proteins are scaffold proteins, high mobility group proteins, HMG proteins, regulatory proteins, all these are collectively called as the NHC proteins. So, remember, when higher level of condensation of chromatin takes place to form the chromosome, then along with histone proteins, certain non-histone chromosomal proteins are also required. Now, when you stain the chromatin, you will see at certain places you will find dark staining and at other places you will find light staining. Why this light staining? Because in this region the chromosome shows loose packaging. Okay? The chromatin shows loose packaging, that is why staining is light, and this portion of the chromatin is transcriptionally active, means it consists of the active genes, because remember, if a DNA has to function as a gene, it should express itself, and you have studied the central dogma of molecular biology. So, for that, first of all, the DNA has to undergo transcription to produce mRNA, and that mRNA will produce protein. So, this is genetically active or transcriptionally active chromatin called as euchromatin, which is loosely packed and light staining, while in certain regions we see dark staining structures. These dark staining structures are places where there is dense packaging, very dense packaging of chromatin, and this portion usually does not undergo transcription. So, this is transcriptionally inactive or genetically inactive part of the chromatin. Okay, we said that nucleosomes in a chromatin look like beads on string structure. You can see here these blue colored structures, they are like the beads, and the DNA in this looks like a string. So, a long string on which at regular intervals beads are present. Okay. So, what happens? The DNA wraps around the histone core, then a regular interval again wraps around another histone core. So, the DNA between the two histones, this is called as linker DNA, means two adjacent nucleosomes are connected through a linker DNA. This forms beads on string structure. So, this is the second level of packaging, but there are additional level of packaging for formation of the chromosome, as we said that at advanced level of packaging takes place when metaphasic chromosome is formed. So, I am showing the entire sequence starting from DNA up till metaphasic chromosome. You see, the diameter of the DNA is 2 nm, which we have discussed. When this DNA is wrapped around the histone to form these nucleosomes, then the diameter is 10 nanometer. This 10 nanometer thread is again coiled to form a structure called solenoid. The diameter of the solenoid is 30 nm, and in one turn of solenoid there are 6 nucleosomes. Okay? So, one turn of one turn curve there are 6 nucleosomes. Then this 30 nm solenoid is further coiled to form 300 nm diameter super solenoid. Okay? And this 300 nm super solenoid is additionally coiled, again coiled, and this forms one chromatid of a chromosome, which is 700 nm diameter, and you know that metaphasic chromosome consists of 2 chromatids. Okay. So, the 2 chromatids, the total diameter is 1400 nanometer. So, you can see how different level of packaging takes place, starting from a diameter of 2 nm up till a diameter of 1400 nm. As a result, a very long DNA, when gets packaged, it is accommodated in a very small space. So, students, we are coming to the end of this lecture now. Let us see what we have done in this lecture. We studied about the structure of DNA and the salient features where it is double stranded, helically coiled, right-handedly coiled. The 2 strands are complementary and anti-parallel to each other, held together by hydrogen bonds, and stacking of the base pairs provides stability. Second thing we said that the total length of DNA in one mammalian cell is 2.2 meter, but the nucleus is very small. How this long DNA gets accumulated in a small nucleus, how this accommodated? For we said that this is possible due to packaging of the DNA, and then we studied how packaging took place, and this is the entire blueprint, that DNA first wraps around a histone core to form nucleosomes. These string of nucleosomes form chromatin, further coiling solenoid, further coiling supersolenoid, further coiling one chromatid, and metaphasic chromosome consists of 2 chromatids. Okay? So, I think you have understood everything. Okay? Revise this and please be safe in your homes. Thank you.